Good afternoon, everybody, and welcome to the next in the series of the British Society for Immunology Connect on Coronavirus webinars. I'm Doug Brown, Chief Exec of the British Society for Immunology. I'm delighted to be delivering this webinar in partnership with the British Society for Immunology West of Scotland Immunology Group. So a fantastic partnership there, bringing, and I will come to her in a second, a wonderful speaker that we're all very excited to hear from. Thank you everyone for joining us again. We have hundreds of you joining us live this afternoon, which is fantastic to see. Of course, we will have the recording of this webinar on our website shortly afterwards in the next couple of days. So please do tell your colleagues that they have another opportunity to watch that via our website. And so it's fantastic to have you all here. And thank you to all of the supporters that are making this webinar series possible. To our existing long-standing journals, Immunology and Clinical and Experimental Immunology, but to our brand new journal that we have just launched from the British Society for Immunology. It's called Immunotherapy Advances and is also supporting this webinar series. And thank you to the support from Miltenyi Biotech as well. Really appreciate everybody's support in making these possible. We have lots more information on our website around supporting all of you during this challenging time in the really important research that you're doing. So please do visit our website, the Connect on Coronavirus hub that we've got. Lots of information on there about how we can support you, the resources that are available, and of course the recordings of all of the webinars that we have delivered to date. That's the BSI. Now I move on to introducing our speaker, and it's absolutely fantastic to have Akiko Iwasaki here with us today, all the way virtually and digitally, of course, from Yale in the US. Who's here to tell us about the immune response to SARS-CoV-2. And there's lots of research emerging every day in this space. So we're very, very pleased to have Akiko with us so that we can hear about the most recent research and understand, really what is happening in our bodies following infection. Akiko will speak for around about 30 minutes, and then we'll have time for Q&A at the end. So please do submit your questions via the question function on the webinar platform, and I will ask as many of those questions on your behalf of Akiko at the end. So that's it from me. Time now to hear from our wonderful speaker. Akiko, I will make you presenter, and then it is over to you.
Thank you, Doug, for your kind introduction. I'm going to share my screen. Okay. Do you see my slide?
Perfect, yes.
Okay, great. So I'm delighted to be here with you today to give the BSI seminar or webinar on immune response to SARS-CoV-2. So I'm from Yale University, and my research is supported also by Howard Hughes Medical Institute. So in the beginning of March, my lab has kind of shifted our research towards COVID-19. We used to work on other viral pathogens and immune responses to other viral pathogens. But when the pandemic hit us, we quickly shifted to looking into immune response in COVID-19 patients in the hope of understanding the basic mechanism of how immune response contributes to both protection and potentially disease exacerbation from this viral infection. So my lab is engaged in multiple different types of research around COVID-19. The talk that I'm going to give today focuses on the top part, which is the analysis of the immune response in humans in COVID-19. And so questions that we address are what immune response features are associated with patients who recover versus who go on to develop worse disease, and if there is any element of sex differences in this immune response. And we've also created a mouse model of COVID-19, whereby we can use AAV to induce human ACE2 in any mouse background, and that allows us to probe genetic link between immune pathways and COVID-19 protection and disease exacerbation. We're also looking at understanding neuroinvasion and neuroinflammation following SARS-CoV-2 infection using brain organoids as well as mouse model of COVID-19. And finally, we are trying to understand the impact of maternal SARS-CoV-2 infection in fetal development and outcome. And many of the studies that we're doing are already posted on preprint archives like bioRxiv or medRxiv. So if you're interested in these other research areas, I urge you to look into that. But today I want to focus on the top part, which is the analysis of immune response in humans. And so in order to get at this issue of how people are responding to SARS-CoV-2 and why there is such variation in the outcome of this disease, we started with collaborators who got together in early March and started a biorepository to collect biospecimen from COVID-19 patients. And so we set up 3 different cohorts. First cohort is the inpatient cohort that I'm going to discuss today. And we enroll, the target enrollment is 400. We enrolled about 300 patients into the study so far, and we collect nasopharyngeal swabs, oropharyngeal, as well as saliva, urine, feces, mini bowel, and so on, in order to understand the link between immune response to viral control in real time. And the second cohort, which we're just beginning to recruit, is the outpatient cohort. And this is patients that have milder diseases, as well as those that have recovered from infection and that have been discharged from the hospital. And again, we are collecting a variety of biospecimen from these patients. And third cohort is also an important cohort in which we recruit healthcare workers. And this served 2 purposes. One is to analyze immune response in response to exposure to SARS-CoV-2 at the earliest time possible, because the inpatient cohort, as important as they are, patients only seek medical care in about 7 days or so post-infection. And so we're missing that critical time window of the first few days of infection. And so monitoring healthcare workers that are exposed to this virus, we're able to capture early phases of immune response. And the second reason we wanted to do this is to implement testing, frequent testing for the healthcare workers, so we can alert them if they do become exposed and infected with SARS-CoV-2. So using this biorepository, we started analyzing their peripheral blood mononuclear cells to look at proportions of different lymphocytes and leukocytes in circulation, as well as looking at T cell activation status and their differentiation status, as well as exhaustion status. And we also looked at cytokines in the plasma in order to understand the systemic role of these cytokines in disease. So the first part of the talk will focus on the longitudinal study of COVID-19, which was just published a couple of days ago. And I want to take you through our thought process of kind of taking these inpatient cohort and comparing it to the uninfected healthcare workers. And what we did is really sort of give a clinical scoring to different patients, all the way ranging from 1 who are hospitalized patients that are not receiving any supplemental oxygen, all the way to those that are requiring mechanical ventilations, as well as, unfortunately, death in some patients. And so we're looking at different severity of infection during the course that we're following these patients throughout in their hospital stay. The first thing we noticed from the flow cytometry of PBMC is that we, as reported by multiple other groups, we also detected decline in T cell numbers in the patients that were, especially in the severe cases. So the green is always the healthcare workers, and the dark navy here is the moderate disease patients who are not on ICU. And then the severe patients are indicated in pink here. And so you can see that T cell number declined in these patients. And conversely, we see increase in monocytes and neutrophils and eosinophils that come up in patients that are infected with coronavirus 2. And we also see in the typical cytokine storm cytokines such as IL-1, IL-6, TNF, and others in patients, and especially increasing levels that are seen during the severity of the disease. And I'll come back to some of these later because they really tell us about the prognosis of these patients. And we also see some of these inflammasome-mediated cytokines such as IL-1 beta and IL-18 coming up. And so what is really kind of going on in the moderate to severe patients? And one of the things that we notice by looking at nasopharyngeal viral load is that, and these are just divided, patient sample divided in days post-symptom onset in 5-day bins. And what we see is that within any given time bin, we don't see a huge difference in the viral titer between these severe and moderate groups. But if you look at this linear regression analysis, what we see is that the severe patients tend to maintain or even have higher levels of nasopharyngeal viral load compared to the moderate patients that ultimately decline their viral load to even undetectable levels. So it may be that the severe disease is driven by a prolonged viral replication in these patients, whereas the moderate disease, these patients are able to control the virus ultimately. And so when we looked at key antiviral cytokines, such as interferon alpha and interferon gamma, what we saw was a correlation, a positive correlation between the viral load in the nasopharyngeal swabs and the cytokine levels for interferon alpha and gamma and TNF and trail, regardless of the disease severity, indicating that it is the viral load that's likely driving the secretion of these types of antiviral cytokines, and further implicates that these antiviral cytokines are not able to control the amount of virus that is within each patient. And moreover, we looked at the cytokines and interferons during the first 12 days of symptom onset in patients and asked what happened to them later on. So we looked at patients who deceased versus who were discharged to their homes. And what we see is that within the first 12 days, the patients that ended up dying of this disease had higher levels of type 1 interferon compared to those that had been discharged later on. And similarly, we see IL-1 receptor antagonists coming up much higher in the deceased compared to the discharged. And similar trends were seen for multiple other cytokines. So what this indicated to us is that within the first 12 days since symptom onset, at least, the higher levels of interferons and IL-1 receptor antagonist, which is really indicative of IL-1 receptor signaling in these patients, are correlating with mortality. And if you look more globally at different cell types and cytokines, what we see is that we see all kinds of cytokines and cell types coming up in severe cases. So these are sort of the type 1 antiviral response, type 2, which is sort of more associated with helminth infection and allergic response, and type 3 is really the antifungal response. And we're seeing elevation and maintenance of all of these cell types and cytokines that belong to all three categories. So for instance, if you look at eosinophil numbers, you see that they become elevated and in the severe cases, whereas the ones that have moderate diseases have sort of a flat line eosinophil numbers. In IgE, similarly, we see them it's coming up in the severe case. IL-4, IL-5, IL-13, these are typical type 2 cytokines that are coming up in these patients. And similarly, these IL-17 and IL-22 are becoming elevated and in a long-term manner in the patients that are suffering from severe disease. And so we wanted to make some sense out of all these cytokines, and what we've done is to do an unsupervised clustering analysis of all the cytokines in all the patients and asked if there's any pattern associated with these cytokines. And that analysis separated the patients into 3 clusters. So the cluster 1 here, and the darker these purple colors are, the more expression there is of particular cytokines. And so what you see is that this cluster 1 has a predominant cytokine in the signature A. And signature A, if you look at the type of cytokines in that group, there are lots of growth factors, PDGF, EGF, VEGF, IL-7, in that signature. And that's enriched in for the cluster 1, whereas cluster 1 patients have less expression of chemokines and type 2 and 3 cytokines and mixed cytokine signature. Cluster 2 patients have elevated levels of these chemokines. They're much enriched in these chemokines, and not so much the type 2 and 3, but also enrichment in the mixed cytokines. In clusters 3, it has the highest level of the signatures B and C, which represents the type 2, 3, and mixed cytokines. And so what do these clusters mean? First, we looked at the age distribution as well as the length of hospital stay between these clusters of patients. And age, there is no significant difference in age between these clusters. There seems to be a little bit elevated median age group in the 2 and 3 clusters, and the length of stay is slightly higher for the 3rd cluster. But what's really segregating these patients is their disease trajectories. So if you look at the cluster 1 patients who had enrichment in the tissue growth factor, tissue repair growth factors, they had a few bumps here and there in clinical score, but most of them actually resolved their disease and were discharged. Whereas cluster 2 and 3 patients became worse and worse in terms of the clinical score. And if you look at the coagulopathy and mortality, cluster 2 and 3 patients had higher levels, indicating that the signatures based on this unsupervised clustering is enough to predict the trajectory of these patients, whether they recover from disease or they become worse in disease. And finally, we also looked at biomarkers that are associated with mortality. And I'm not going to go through all of these factors, but what I want to draw your attention to are these factors that are associated with mortality much above this sort of neutral zone, which is 1. And what these cytokines are is interesting. There's IL-18. Again, this is sort of the typical cytokine released as a result of inflammasome activation. We also have interferon alpha as a very predictive factor for mortality, and IL-10 and neutrophil levels and so on. So this again indicates that even though interferons are absolutely important for antiviral defense, in the case of severe COVID, we're seeing elevated levels in patients that are associated with mortality. So what we've learned from the longitudinal study is that persistent viral load is detected in the severe cases, and this viral load is actually driving inflammatory cytokines and chemokines and interferons. And this increase in interferon alpha and inflammatory cytokines within the 12 days of symptom onset, which translates to perhaps 2 to 3 weeks post-exposure to this virus, is predictive of mortality. And there are 4 signatures of cytokines that cluster patients into 3 different clusters, and those that developed tissue repair type of signature do well, whereas others who develop more mixed cytokine signals do poorly with disease. So what this tells us is that this viral infection is likely inducing the kind of infection that has either very low or delayed interferon response that's leading to viral persistence and inflammation, whereas interferons are playing a more pathologic role rather than antiviral role, especially during the late stage of infection. So learning from the longitudinal study, we wanted to analyze some host intrinsic factors, such as sex, in determining the kind of immune response that develops against COVID-19. So the impetus for this came from the fact that there are multiple reports from various organizations showing that male sex is a risk factor for severe COVID disease. So in US, about 54% of the COVID-related deaths are in men. In China, similarly, there's a higher level of men who succumb to this disease. And even the hospitalization rate are higher for men in China as well as in Italy. And this isn't the first time there's a sex difference in infectious disease. There's been multiple studies done by multiple groups that have indicated that there are propensities for having either a female dominant or male dominant prevalence as well as severity of diseases. So, interestingly, the MERS-CoV, which is a cousin of SARS-CoV-2, has a prevalence or incidence as well as severity in men as well. So there might be something common between the beta coronaviruses that we're seeing this male dominance in disease severity. So we designed the study in order to analyze baseline immune status in men and women who are coming to our hospital at the very first time point. We collect their samples, and we excluded patients that were receiving tocilizumab, which is an antibody against IL-6 receptor, as well as high-dose cortisone treatment, because we wanted to monitor their immune response in the natural form that is not being affected by any of the immunosuppressive agent. And so we took these patient baseline samples, which ended up being 39 patients. And we also recruited healthy healthcare workers, as I mentioned before, as controls. And we also extended this analysis to 98 patients at all stages of disease after we had learned something from the baseline immune characterization. And so first, this baseline cohort, when we look at the nasopharyngeal viral load or the saliva viral load, there was no significant difference between the male and female patients. So the male is always orange and females are always purple. You can see that there is a huge variation in the viral load at the first sampling. Of course, all of the patients were SARS-CoV-2 positive at the time of admission, so they were all positive to begin with. But when they enrolled in the study, this is the distribution that they had. And then antibody response against the SARS-CoV-2 spike protein, IgG and IgM, were also not significantly different between the sexes. However, we did see some chemokines being quite different. Well, first of all, in both sexes in this case, you see that these are the healthcare worker levels here, and the ones that are COVID patients are indicated on the right, and you see that they're much higher levels of CCL5 and CXCL10. And CXCL10 is one of the most upregulated chemokines that we find in COVID patients in both sexes. However, there were some cytokines and chemokines that were elevated more specifically in male. So this is IL-8, an important chemokine for neutrophils, are elevated in male compared to female. And similarly, IL-18 had higher levels in male compared to female. So there are some pro-inflammatory cytokines that have male propensity. Whereas if you look at their the peripheral blood cell types, non-classical inflammatory monocytes were elevated in males compared to the female patients. And moreover, if we look at the correlation between CCL5 levels versus the non-classical monocytes in their blood, we see a positive correlation in the male patients only between these two factors, whereas there is no correlation seen in women. But conversely, when we looked at activated T cells, they were more elevated in female patients compared to male patients. Here is a CD38 positive, HLA-DR positive CD4s and CD8s, and both are elevated in female patients, and also PD1 positive, TIM3 positive terminal effector type of cells are also elevated in female patients. So we've now detected sort of pro-inflammatory cytokines that are elevated in male patients and activated T cells that are elevated in female patients. And can we use these data to tell us about the future disease trajectory? And so we took these patients' baseline samples, and then we analyzed whether there's any difference in those who did poorly with disease, which is the filled circles here, and those who maintained or stable disease, which are the open circles. And what we see is that the male patients who developed worse disease were higher in age or older, whereas female patients, we didn't see a difference in the age distribution. And similarly, the BMI was higher in the male patients that developed worse disease, whereas there was no significant difference in the female patients. And I mentioned to you that male patients tended to have lower T cell activation than female patients. And if you look within that male patients who developed very poor T cell activation, they are the ones that ended up getting worse disease here, as well as interferon gamma secretion from T cells were much lower in male patients that went on to develop worse disease outcome. Conversely, in the female patients, even though many of them are developing really good activation of T cells, if they tended to express or secrete more of these pro-inflammatory cytokines and chemokines, they belong to this group of female patients that developed worse disease outcome compared to the stable counterpart, whereas there was no such difference seen in the male patients. And what's striking to us is that when we looked at the age distribution and the activated T cell phenotype here on the y-axis, or the interferon gamma secretion from CD8 T cells, what we see is that there is a decline in the men's ability to stimulate activated T cells as they age, whereas in women, there was no decline. In fact, if anything, it's kind of still going up as women age. So this told us that for this virus, women maintained T cell immunity even in older age, whereas men failed to do so. So to conclude this part, we saw that men developed more inflammatory cytokine responses than women at baseline, whereas women developed more activated T cell. And men who developed very poor T cell immunity tended to do worse with disease, whereas women who developed more pro-inflammatory cytokine had worse disease trajectory. And finally, women aged better with respect to their T cell activation than men. So I want to end here by thanking everybody who was involved. This is unfortunately not the entire list of people who contributed, but I just want to highlight the impact team. This is the team that came together in early March. It set up the biorepository, and they include many people from the School of Public Health, Nursing, Global Health Institute, and virology, and so on. So, and immunology, of course. So this is a very, very important group that was set up that enabled us to do this analysis. And also my lab members, as well as the COVID funding, which came through very rapidly in order for us to be able to carry out this research. And I will end by thanking my amazing lab members who stepped up to the challenge immediately after the pandemic hit us in the U.S. And we meet every week on Zoom to discuss about research and how we can implement more testing. So thank you, and I'll end here for questions.
That's fantastic. Thank you very much, Akiko. A wonderful presentation there, and you'll be pleased to hear we've had lots of questions coming in. So I've tried to group them and theme them around different through the number of questions that are coming in. So bear with me and I will ask you some of those now. So first one was around cytokines increasing but don't seem to have that effect on viral load. One question was around, is there a difference in cytokine receptor expression? So for example, do the immune cells become ignorant to the cytokines that are flowing around?
Right, so that is also a very puzzling thing that we've observed. You know, normally you would think that the more type 1 interferon there is, the more antiviral effect there should be. And we also detected interferon stimulated genes also upregulating. So it's not that cytokine responsiveness is all gone, but rather this virus is able to evade the effect of cytokines and interferons. And if you look at the literature from SARS-CoV-1, there are multiple genes that virus encodes to interfere with every process of interferon stimulation as well as receptor signaling. So we suspect that that is what's going on, is that the virus is interfering with the signaling through these receptors.
No, I mean, really interesting. Do we know what the root cause is of the cytokine storm? I mean, is it the virus having that effect? I mean, do we know what specifically is happening in the body?
Well, so we only were able to monitor peripheral blood in this study, so we can only sort of speculate and deduce from that. But essentially, what we saw was this very tight correlation between the viral load and the level of the cytokines. And so the virus is ultimately driving the cytokine storm. But what we really don't understand is what's happening at the real site of disease, such as, you know, deep down in the lung or in other places like the gut, where this virus is also replicating very highly. So we don't have tissue level information yet, but I think that's something we have to do in the future.
Oh, definitely. And those samples will be coming through to labs, I'm sure, as we are speaking right now. There are a couple of questions around T helper cells, and do you have any insight into how the different subsets of TH cells might be having a role or affected in COVID?
Right. So as I mentioned, there is this severe lymphopenia, T lymphopenia, that happened in these patients. And so even though T cells are becoming activated, the vast majority of them are being depleted from, at least from circulation. So whether those cells are having a huge impact on cytokine storm, for example, we don't really know. It's unlikely that the T cells are the source of cytokines in this case. It's likely macrophages or some other plasmacytoid dendritic cells or some other cell type that is responding to virus, whether they're infected or they're just taking up these viral particles, is likely the source of the cytokine storm. And yeah, the T helper cells, as I mentioned, all kinds of T helper cells are being activated. So, you know, what they're doing and whether they're maladaptive for the viral infection, it's something that we're working on.
Brilliant. And it was a really interesting question that came in asking whether you're aware of any studies in HIV patients where, of course, immune cells are certain subsets are lacking in HIV patients. And is there a protective element to severe disease in HIV patients? Are you aware of any literature that's come out looking into that?
Right, so there's been a few reports on HIV patients, and what they are kind of reporting is that surprisingly, they're not getting a severe COVID disease. Most of them are sort of having a normal course of infection. And so why would that be? That's a really key question, like whether T cells, even if they're there in low numbers, may be able to perform the function that's needed for countering this virus infection. And so this is a very big puzzle right now, like how the T cells that are being affected by HIV is able to still pull through to protect these patients.
No, definitely. So much more we still need to know, isn't there? So there were quite a few questions coming in around length of immunity. Do you want to, if you're able to elaborate on what you've seen coming from the literature, from your own studies, and thoughts on the length of that immunity and likelihood of reinfection?
Yeah, so that's a major focus of attention from many people, including immunologists. And so, yes, there have been several studies published that demonstrate decline in antibody levels and neutralizing antibody levels in patients within the first 2 to 3 months after their recovery. And yeah, so obviously, you know, it's not like the measles virus where once you're infected, you maintain high titer of IgG for the rest of your life. This virus appears to only induce a transient antibody response. However, this may not be a huge cause of alarm, because even in those patients who have undetectable levels of antibodies, they still would have memory B cells and memory T cells that can respond quickly during second exposure. So hopefully they can sort of, you know, clear the virus much more quickly and have very low symptom, or probably no symptom from the second exposure. However, you know, we just have to see. We don't know enough about the immunity to this virus yet to say one way or the other, right, whether immunity lasts in people who are previously infected. So that's something that we're also trying to follow up. It's still very early in pandemic. It's unclear whether reinfection is going to be a big thing coming up.
No, yeah, I mean, a very important point there. It feels like we've been living with this for years, hasn't it? But no, it's still very early on. And as I say, lots more to find out. I mean, your research is absolutely fascinating. I mean, is there anything from emerging from your research or your colleagues' research that you think we need to make sure we join up more with the vaccine development efforts that are happening globally? I mean, what are you seeing that's coming through that actually would influence, would enhance, would narrow down the potential vaccine candidates for us to identify one that might work?
Right. So today I mostly focused on what happens when once you're infected. And what happens in severe cases is really alarming because there's all kinds of inappropriate immune responses that are becoming stimulated in a very chronic way. And so the long-term consequences of this type of disease is something that worries me a lot. Even after discharge, these patients are having chronic issues with their health. And so vaccines is really the only way to prevent, you know, this disease from happening, and also to confer herd immunity. And so, but whether our insights for the natural infection and immunity provides a basis for a better vaccine, that's unclear. I think so. Before I started COVID research, I was working on vaccine approaches to viruses, and from that, reading the literature, I think the coronaviruses are not a huge impediment for vaccine development. They don't have a mechanism to evade antibodies, for example. And so I'm hopeful that vaccines that can generate pretty good neutralizing antibody titers and T cell immunity are going to protect from at least the disease from COVID-19, if not infection altogether.
Brilliant. I'm sure we are all very happy with that message of hope, Akiko. A couple more questions before we let you get back to your important research. Focusing around treatment, so moving from vaccines to treatments. In the UK here, recombinant interferon beta is being trialled as a potential treatment in the UK. May also be the case in the US. And some of the early data coming out of that look like it's actually quite promising. How do you think that fits with the correlations you've seen with your studies of interferon levels and link to mortality?
Yeah, so that's why I put up that diagram to emphasize that what we are looking at in our study is very late phase interferon. So, likely 2 to 3 weeks post infection. And that's been known from SARS and MERS research that late onset interferon is, toxic to the host. Whereas what you're talking about, Doug, is the early treatment with interferon beta, or even prophylaxis prior to exposure. And I think that holds much promise, because what that does is to kind of prevent the viral infection and replication in the very early phase of transmission. So I think that is a really good thing to do. But what we want to be careful is not to start the treatment too late, because that's where we're seeing this correlation between interferon and toxicity.
That's great. That's great. And final question for you. Clearly, there is from your research showing that there are sex differences between male and female in the responses, with the female response seemingly better and reducing that rate of severity and mortality. So one question came in asking whether convalescent sera should only be used from female donors. What are your thoughts on that?
Yeah, well, so the convalescent sera, we currently don't understand what exactly the level of antibody needed to protect, either uninfected or infected people from COVID disease. So, but, obviously, we all believe that certain levels are needed. We didn't find a huge difference, actually significant difference between male and female antibody levels. So I believe that as long as a male or female is developing good enough antibody, they should be able to donate their plasma for therapy. So I want to make sure that that message is clear, because I don't want, men to start thinking that they cannot donate their blood.
Great. Wonderful final message there, Akiko. I'll leave the questions there, Akiko. What a fascinating, interesting, fantastic talk you've given us, at least here in the afternoon in the UK, clearly morning in the US still. And so you've got lots to be getting on. So we're going to bring the webinar to a close. Everybody who has been watching, and hundreds of you have been here, as I mentioned before, we'll have a recording of this webinar online soon. So please do, if you want to view it again, do that, or promote it to your colleagues who are unable to join us today. And we have more to come as well. Again, information on our website, information via our social media channels, and through our emails. We have more webinars in the pipeline that will keep your appetite, your thirst for this research quenched, hopefully. Don't forget, the British Society for Immunology is here to support you. So please do visit our website. Please do get in touch if there's anything more you feel we can do to support you in your important research at this point in time. And really, that just leaves me to thank all of you, our viewers, to thank our supporters, the 3 journals, Miltani Biotech, and most of all, thank you, Akiko, for a wonderful presentation, really fascinating, and we've all taken a lot away from this. I can tell number of questions is usually a good indicator of engagement, and they're still coming in fact. Sorry, no more time for questions. So thank you very much, and thank you everyone who's joined us. We will see you next time. Take care.
Thank you very much, Doug. Thank you.